We're very pleased today to welcome Dr. Anath Shalev. She's an international expert on the molecular biology of diabetes. Her research centers on beta cell biology, apoptosis, oxidative stress, transcriptional regulation of gene expression, and development of novel therapies for diabetes. Her laboratory identified thioredoxin interacting protein, or TXNIP, as a therapeutic target that can be used to block the death of pancreatic beta cells and preserve a diabetes patient's ability to produce insulin. Dr. Shalev holds the Nancy R. and Eugene C. Gwaltney Family Endowed Chair in Juvenile Diabetes Research in the UAB, Hersnik School of Medicine, and currently she serves as the director of the UAB Comprehensive Diabetes Center and as a professor of UAB Department of Medicine's Division of Endocrinology, Diabetes, and Metabolism. Today, she is coming to us to talk about Tiximed, which is, they are developing the first oral non-immunosuppressant treatment for people with type 1 diabetes and type 2. And we're very happy to welcome her today and have her present what's going on with the company and the molecule, and interact with our academic and clinical audience. Please feel free to drop questions in the chat during the talk, and we will field them as we go. Thanks so much for joining us, Annette. The last time we spoke was March 22nd of 2022 at the Sugar Science, but we also went to your IDS presentation in 2023 where there was a lot of buzz around verapamil, and we're really excited to hear what's going on with your next sort of venture here.
Great. Thank you so much, Monica, and thank you so much for inviting me to talk to you kind of as a follow-up to what we have done. And today I'm kind of talk to you, put a different hat on. I'll talk to you from the Tiximed side, which is a startup that we started from UAB, and where I'm a co-founder and the chief scientific officer. So just kind of for to make that clear that we have that going on here. So again, we founded Tiximed in order to develop and commercialize a TXNIP inhibiting medication as a first of its kind oral therapeutic for type 1 and type 2 diabetes, as Monica just mentioned. And just to give you kind of an overview of the team, I collaborated with Michael Goodrich to have the financial aspect and the business aspect, and we have a great scientific advisory board. Many of you will know many of them, with David Harlan, Alan Moses, and Ron Kahn. And so they have been extremely helpful in those endeavors. Again, coming from a university startup, a lot of the development for a new drug we could not do. And so we worked with experts all across with those CROs. One in particular, Kinexum, helped us along with the different ones to do all the safety and all the pharmacology and chemistry development, manufacturing, and all that stuff. So that's been a great help to have a great team on board, and a very steep learning curve for those that want to kind of go out on that direction, but very exciting, of course. I don't think I have in this group to talk a lot about the problem. Again, those slides are more kind of for a general audience, but I just wanted to kind of give people a background, and so we can go more into the discussion. Obviously, diabetes is a major problem. It affects many people. It affects different organ systems, and getting better control would be really huge, both for type 1 and type 2 diabetes. Unfortunately, for type 1, there's even less development in terms of targeting some of the underlying causes of the disease. And of course, it also is a very big economic issue. So just to give people, again, the background on the target that we're trying to address here, thioredoxin interacting protein, or TXNIP. It's involved in oxidative stress. So when it's increased, oxidative stress in the cell goes up. And it's extremely well conserved across species, and that helped us really develop this as a drug target. We initially identified it as the top glucose-induced gene in human islets that we had exposed to high glucose, and it really was the one that stood out. Importantly, it is not just in vitro when we expose cells or islets to high glucose that it's increased. It's also increased in all the different diabetes models that we and many others have looked at in terms of rodent studies, in terms of mice, but also in people with type 1 diabetes and type 2 diabetes. In addition, what a lot of other groups have shown, not us, very nice work in terms of the TXNIP effect on inflammation. So it's not only oxidative stress, but also inflammation, partly through this inflammasome activation. And there's been a lot of work going on in that direction. So also from that aspect, something that makes sense that is involved in diabetes. And then it has detrimental effects on cell function and survival. And as you can imagine, particularly in the beta cell that is more prone to oxidative stress, the increase in TXNIP has more detrimental effects, but it does also have detrimental effects in other organs, such as the heart, the kidney, the retina, and even in neuropathy. So all these other systems are affected by the increase in TXNIP in the context of diabetes. And many different groups have shown the detrimental effects and the beneficial effects of downregulating TXNIP in those tissues. And as you can imagine, when there's an increase in TXNIP due to the diabetes and the elevated glucose, that then causes beta cell dysfunction, beta cell death, and you get into the vicious cycle of worsening of the hyperglycemia, and that's shown kind of in the middle part. Now, again, the rationale to target TXNIP from a preclinical and clinical aspect is what I just mentioned before, that TXNIP is increased in these different models and in humans with diabetes. And then we had shown extensively that genetic TXNIP deletion protects multiple mouse models against diabetes without causing any adverse effects. And other groups have shown this in other models as well. In addition, the non-specific pharmacological inhibition of TXNIP with verapamil, as Monica just mentioned, improves diabetes in mice. Initially, that's how we found it back in 2012, but then also in humans with type 1 and type 2 diabetes. And so we had the initial study, small study that we published back in 2018 in Nature Medicine, where we saw that adults with recent onset type 1 diabetes fared a lot better when treated with verapamil as compared to with placebo, meaning that their C-peptide area under the curve was a lot better, showing improved beta cell function over the year, and then also together with that, an improvement in the sense that they required less of an increase in the required insulin in order to maintain glucose control, and a larger time period spent at target. And then we did a follow-up study in 2022 where we showed that those beneficial effects were maintained when people continued to take verapamil for at least 2 years. On the other hand, the group that stopped verapamil, they worsened. So you have to continue to take the medication, but those benefits are sustained. And that makes sense, obviously, with the way we know that it functions. And then excitingly, just last year, Forlenza et al. Showed in the CLEVER study that the beneficial effects of verapamil are also shown in kids with type 1 diabetes. So it's not only in adults. And so that was a nice validation of that work. And so with this, we believe that targeting TXNIP is a novel and disease-modifying approach for the treatment of diabetes, and we have proven with verapamil that it is translatable to humans. So what we've seen in those mouse model, in that particular case, with this particular target, again, it is very well conserved across species, is translatable to humans. However, of course, verapamil is a calcium channel blocker, and with that, it's not specific by any stretch. And so we obviously wanted to have something a lot more specific. One problem that verapamil can cause as a calcium channel blocker is that it can cause arrhythmias, particularly atrioventricular blocks and heart blocks, as well as hypotension, low blood pressure. We didn't see much in our small study, and neither did the CLEVER trial, but there's larger studies are ongoing currently with Inodia in Europe. And so we'll have to see how that works out. For that reason, people have to be checked with EKGs before started and also throughout to make sure that they don't develop that, because some people are just more susceptible to those side effects. But that obviously limits its use. So wanting to have something more specific, we developed this new molecule that we call TIX100. That's based on a high throughput screen and extensive medicinal chemistry to optimize the structure. And the hit that we ultimately came up with is this TIX100 that I'm going to talk to you a little bit more about. And so here you have the preclinical proof of concept of this TIX100. We used the same models that we had shown worked to translate rat-mill from the animal models to humans and tested TIX100 pretty much in analogy in those mouse models. And so we saw that TIX100 improves glucose homeostasis in non-obese mice and genetically obese, insulin-resistant, and diabetic DB/DB mice, a very severe form of type 2 diabetes, and also in streptozotocin diabetic mice, and even does so when started after the onset of overt diabetes. And so it also has beneficial effects in human islets. So together, that's what was very promising to us. And we published initially those preclinical studies in Cell Metabolism in 2020. Now, TIX100 has several safety features that we liked a lot when we started studying it. So, as mentioned, unlike verapamil, TIX100 is not a calcium channel blocker, and as such, it does not have the associated potential for these cardiovascular side effects and the heart block. Also, TIXNIP deletion in animal models and TIXNIP inhibition with verapamil in humans has so far proven to be safe, including in adults and in children with T1D. And most importantly, the therapeutic goal, and this is really critical to realize that and to remember that, the therapeutic goal is to normalize the pathological elevation of TIXNIP back into non-diabetic values. And so we're not trying to knock out TIXNIP or to make it, the levels go down to zero, but rather just bring it down to normal non-diabetic values. And so we have actually a couple of interesting features that help de-risk. If you think about the risk of always when you're trying to translate something and develop a new drug, there's always a lot of those things fail. But in this particular case, at least several of those questions we were able to address and give us confidence that this will actually work. And most recently, actually, the biggest achievement that we have right now is that we just got last month, we got IND approval from the FDA for TIX100 to proceed into clinical trials. I get to you in a moment for what that all requires. Now we have the peer-reviewed preclinical data. TIX100 specifically targets TIXNIP, and that has already been translated, again, as I mentioned before. It is a lot more potent. It's also more effective and more specific, and I don't have the data to show you here, but I just wanted to mention that. And it's actually more animal models by now, that including human islet studies, that have shown the efficacy of TIX100. And then, of course, it has also a favorable pharmacokinetic and safety profile. That's how we initially selected it. But since then, of course, in order to get IND approval, it went through extensive, actually almost 3 years worth of extensive studies to show its safety, to show its pharmacokinetics, to also do the whole chemistry and control, and then the manufacturing. So we were able to manufacture, increase the manufacturing from just milligrams to grams and then to kilograms. And we have now also did drug product development with excipient compatibility and all that stuff. And so we have currently capsules ready to go into the clinical trial. And then an additional kind of tidbit of potential promise that TIX100 holds is that in addition to its effects on type 1 and type 2 diabetes, at least based on the preclinical studies that we have published and that we actually have ongoing as well, is that it also helps in muscle D, so in fatty liver disease. And so that is an additional feature that down the road may become attractive. Now, the target product profile that we want for this new drug for TIX100, again, the mechanism of action I kind of mentioned to you, but just to kind of as an overview again here, you have TIX100 working on the beta cell where it inhibits TIXNIP that helps with TIXNIP blocks insulin production, so we have an increase there, blocking the oxidative stress, beta cell death, and the inflammation that I mentioned to you. What I did not show you, but it's in the paper, in the cell metabolism paper, TIX100 also has a clear inhibitory effect on TIXNIP in alpha cells, which verapamil does not. So that is a big advantage from the TIX100. And it leads to inhibition of the hyperglucagonemia, but really only of the hyperglucagonemia. It does not inhibit glucagon secretion in response to low glucose or to any stressors. So with that, the liability for hypoglycemia is very low, and we tested that also in vivo. So even when you make mice hypoglycemic with insulin and you give them TIX100, they're able to sustain their glucose levels as well as the ones that are just getting vehicle alone. And so in a way, it targets the problem from two different angles. From one, on the beta cell side, it improves beta cell survival and function, but then also from the alpha cell side, which has been known for a long time, but it has not been drugs to address that had not been successful for a number of reasons. But it also improves this hyperglucagonemia, and with that glucose homeostasis, because when you limit this excessive glucagon secretion, you also block the excessive hepatic glucose production that worsens the hypoglycemia. So obviously those things, those mechanisms and processes are in play in both type 1 and type 2 diabetes. But again, I think in type 1, where the biggest need is, that's where it's going to have the initial major impact. And so
Anath,
again,
could I just ask one question?
sure.
With TIXNIP, it's in the nucleus under stress, it moves into the mitochondria, and you kind of unpack that mechanism in your paper. But in the alpha cell, can you kind of talk a little bit more about how TIXNIP is active in the alpha cell and gets in that? It sounds like it's almost like a regulated change there.
Oh, yeah. Well, so I mean, it's a very good question. There's still so little known about how the alpha cell really works. So we haven't looked specifically whether it works any way differently from our. I mean, it blocks the expression and the transcription of TIXNIP in the alpha cell as much as it does in the beta cell. So that part is the same. The big question for us was initially, how come is it really TIXNIP that blocks the glucagon secretion? So that was actually, even when we published initially the cell metabolism paper, we weren't 100% sure. It looked like it because we were able to mimic it by in vitro by knocking down TIXNIP, but we weren't sure. But then we actually generated an alpha cell specific TIXNIP knockout mouse, and the islets from those mice, they also have the same phenotype of reduced glucagon secretion under high glucose conditions. So we do believe that it is mediated, that effect on the glucagon secretion is truly mediated by the inhibition of TIXNIP.
Interesting. And then I just wanted to ask, sort of while we're on this slide, what about the other cells in the islet? Are they also are they experiencing changes in the presence of TIXNIP?
Great question.
TIX100.
I hope you're one of the reviewers of our R1 that we just submitted.
Well, I'm not sure about that, but yeah.
Yeah, no, absolutely. Yes, we do see in other cells as well, and it's very interesting because they're both verapamil and TIX100 have effects, but they're different, and so we're trying to untangle that web, and it's very, very interesting what we're finding with this.
Yeah, well, that's great. Everyone's going to have to keep an eye on that horizon.
Any other questions while we're on this?
Okay, just listening. Okay, let's keep going.
Okay, I'll keep going. And I'm not going to go through all these attributes here, but again, it's just kind of in general, of course, we want to target the general things that we want to improve. Of course, there's this issue with the FDA, what is considered an endpoint, and so we're trying to work on that as well. But we're hoping, of course, that this is considered an adjunctive therapy at this point. We do believe, based on our experience with verapamil and this being more potent, we do believe that this will help reduce the dependency on exogenous insulin, and with that, reduce then the number and the frequency of injections, or the amount of insulin through pump infusions. With that, you obviously also get automatically the improvement in hypoglycemia, so less risk of that. And we also do believe that with the additional effect on the hypoglucagonemia, you also improve the need, you reduce the need for insulin by not having this excessive glucose production. The other thing that we do believe with this is that we're targeting at this point, similar to verapamil, stage 3 at this point. But of course, with anything that addresses beta cell health, the earlier you start, the easier it is, and the more you can preserve. So it definitely would also be attractive for earlier treatments, stage 1, stage 2, to prevent the progression of the disease. And in the case of TX100, because of the effect on glucagon and those resulting effects on glucose control, we do believe that it could be even effective even in the later stages. So not just in the new onset, but even in people that have very little or no insulin of their own, that by itself could also help with the glucose control. And again, being an oral medication, very easy to combine with a lot of the other drugs. And that's another important point that I want to make, maybe while we're on this slide, is that since the mechanism is very different, since it's targeting TXNP, which other medications do not, it lends itself to combination therapies, of course, with any other of the medications that are out there for type 2, like GLP-1s and SGLT2s, etc. But then also in type 1, if you think of some of the immunosuppressive regimens, or actually immunomodulatory, hopefully, regimens, that might be helpful to combine with something that targets particularly beta cell health, but then also initially blocks the immune attack.
Yeah, I mean, I thought immediately of teplizumab. So,
Exactly.
right.
Exactly.
So, and when you imagine that, you think, okay, well, that course is like what, a 2-week infusion, and
Right.
daily infusion. So, so that would come first for those maybe in stage 2 or something, and then you'd get
Yeah.
on this regimen of
Exactly.
of
Exactly. With something with that
100.
helps with beta cell health. Exactly.
And then we've got a question from John Callahan. Will the phase 1 study arm include people with diabetes stage 2 or 3, or healthy people?
Well, so the phase 1A is, of course, initially is just a safety and tolerability. However, we are planning already to have patients with established type 1 in our phase 1 trial. So that is factored in our plan. Yes.
Sort of tacking on to this, this idea of dual therapies in the prodrome, or the lead up in the early stages of type 1. So TXNP is a vitamin D upregulated protein, right? It modulates cellular redox balance. But so is there any need once you are on the medication, or once hypothetically people are on the medication, to reduce vitamin D in the diet or
No,
modulate that? Because it's just
no,
not
no.
responsive.
No, it's actually a little bit of a misnomer. It was in the name came originally from the fact that it was a erythromyocyte cell line that was differentiated with vitamin D, and at that point, TXNP went up, and so it had more to do with the differentiation of the cell, that only when it was differentiated it expressed TXNP, than with any major effect in terms of vitamin D kind of pathway or signaling pathway. So there's really not much in terms of vitamin D and TXNIP other than the original discovery.
Oh, great. That's a good clarification. Thank you.
Sure. Any other questions right now? No, not I'll continue.
Nope, crickets.
Oh, yeah, there was.
Nope, nothing
No.
going.
Okay.
Nothing happening.
Okay. All right. So, and again, I don't think I have to tell this group why we want to focus first on type 1 diabetes, but obviously there's currently no oral medication available, and so we want to do it for that reason. We kind of had that paradigm shift that we believe that it's not, it's of course it's an autoimmune disease, but we have to take at least a two-pronged approach in order to be successful, tackling the autoimmunity, but also the beta cell biology. And so we have kind of the edge here because it seems that that has not been kind of a focus as much as the autoimmunity has. And of course, when you focus on something, and again, the plizumab is a good example, you get somewhere ultimately. So we're hoping that this would kind of be the other side of the coin. And also in terms of practicality and in terms of the bringing something to the clinic, it requires a lower number of patients when you do those studies in type 1, a shorter length of the trials, and as opposed to type 2 diabetes. And then, of course, the nice thing is when something, in this case, that targets beta cell works in type 1 diabetes, it will work also in type 2 diabetes, which, by the way, was also true for Rapmil. But the other way does not typically work. So we're excited about that. But that's why we're focusing on type 1. And so where are we currently? So as I mentioned, we've done all this chemistry, manufacturing, and control of CMC, manufactured over 2 kilograms of the medical grade material with Atiximab, and this was done with again help of CROs. We did the drug product development. We have 4,000 capsules ready to go. A lot of toxicology and pharmacokinetics. Those are very long studies and expensive ones. We have now the active IND that we were excited about. And have the clinical CRO lined up to do it. And I don't want to go into the finances, but again, that was what we were working towards for the last 3 years, is to get that FDA approval. And I think that's kind of where I'm at. And so we can stop here, and I'm happy to take any questions.
Yeah, it's a fantastic progress. Kudos to you for you really
Thank
pushing
you.
this forward and continuing to push it forward. I know many people are watching to see, how this progresses. And so let's open it up to questions. Let me see if I see a couple of industry people here. Okay, I have a couple questions. So if we talk about, how might TXNIP administration affect future viral infection of beta cells? So for instance, if this response pathway is offline, what are the outcomes for alpha and beta cells if they do become infected in the future?
With TX100, we have no indication that it affects, any infection or anything else at this point, but we of course don't know. We have not tested that.
Yeah, because it interacts with the NLRP3 inflammasome to get the cells.
Right. But again, I think that there again, I think the important thing is that we're not eliminating, and I did not show the data, but um, when we were developing this, the chemists were getting very upset because they said we're not getting 100% reduction in TXNIP. We actually we're about 50% of what we can lower, and no matter how high we go, I mean that pretty much it maxes out at about a 50% reduction. And again, they wanted to get 99%, but we were actually quite happy with it because we saw that is totally sufficient in order to have the beneficial effects. And it adds an additional safety feature where you don't get to the very low levels. So we're not really concerned about, are we setting them up for a higher level of infection? In addition, and I should mention that there's obviously there are mouse models that have no TXNIP whatsoever, so they're whole body TXNIP deficient mice. And they don't have a higher rate of infections. They don't have a higher rate of leukemias or any of the stuff that you would expect with those things if there were an issue. The only thing that they do have, and particularly the males, is that they're less tolerant to starvation. And so that's what we're thinking. This is kind of a thrifty type gene that in the old days it was really protecting against infection and all the microbes that are around it, and against starvation. It was shutting down the beta cells in order to not die from hypoglycemia. But now, when there's an excess in food and energy, we don't really need it. So I don't think at this point that even if we were to reduce it more than what TIX100 currently can, we don't expect necessarily any detrimental effects, and that's another nice safety feature that we have with this.
Yeah, very nice. And really, it seems as if it's not really an on-off switch. It's a more of a tuning situation.
Exactly. That's exactly right. And so the goal is really just to bring it back down into the non-diabetic level, or to prevent it from actually going into the diabetic level. So prevent kind of the external factors to cause the increase in TXNP that then starts the whole vicious cycle. If you just can block that, don't have to even reduce it below that, you have the beneficial effects.
Yeah, that's really nice. And it's great how you really, kind of you went for these top glucose increased genes, and then from there you really followed the trail of mechanism into the drug development stage. I wanted to see if you might talk a little bit about the Enodia partnership and how they're being very helpful, especially for other U.S. Type 1 diabetes focused bio entrepreneurs, because I think that that's really been helpful in your partnership, right, Enodia?
Well, we have not actually worked with Enodia at all, personally. So when they have not been involved in any of our studies directly, it's just
Okay.
nice to see that what we have started, that they're bringing it to another level with a very large multi-center trial in a different population,
Okay.
taking it outside of the US. And so I think that's been really great. We're obviously very open to new partnerships, and they want to. We're very happy to work with them.
Yeah, no, I love that collaborative piece, and I think that they've been very proactive in kind of bringing clinical trial participants together.
Absolutely. And they're also doing some of the combination studies of Rapmil with the immunomodulators. So I think that is, hopefully we'll learn a lot from those studies as well.
Yeah, Chantal Matthews' leadership has been very
Oh, it's
instrumental
fantastic.
pushing
Yeah.
things forward.
Mhm.
Okay, we're just at the top of our, or the end of our hour, a half hour here. Last call for questions. Okay. Well, thank you so much, Annette. This was really great. It was a really nice chance to catch up and see where you've gotten to in really less than 1 year. We saw you last May at the IDS, and not even a year has passed, and you've really made some important and incredible strides. So thank you so much for all you're doing, and we cannot wait to see what happens next. You have a lot of congratulations in the chat and great work. Best of lucks. So
Well, thank you all, and thank you, Monica, very much for having me.
have a great rest of your day. Bye bye
You
now.
too. Thank you. Bye-bye.